and this is your biology lecture. Okay, now we've completed the transport in animals. We did the whole circulatory system, blood, heart, lymph, and so many other factors related to the circulation in animals, right? Let's just have a brief lecture about how transport is occurring, because this is the transport system chapter, of exactly how is transport occurring in plants. Okay, so let's begin with today's session on plants only. Alright, so we begin. Now, first of all, as you all know that transportation in plants happens in a different vessel for water and in a different vessel for its food, right? So you have the xylem and then you have the phloem. We've been knowing this since ever our school days. Now, what is it that xylem is going to be conducting? We all know that xylem is responsible for the transportation of water, whereas when we talk about phloem is responsible for the transportation of all the sugars which are produced in the plant, right? So now, there's one thing that you need to know before we begin this lecture, and I just recall this, okay? When we talk about xylem and we know that it is responsible for the transport of water, well, where is that water being absorbed from? That water is being absorbed from the roots, and the water is going to be, or rather the roots are responsible for absorbing the water from the soil, and it is those very same roots which are going to push that water above so that all the remaining parts of that plant may receive it. This is all being done through the vessel which we call as the xylem. But on the other hand, when we talk about the phloem is responsible for the transport of the sucrose, or the transport of the sugars produced by the plant. Now, where are the sugars produced by the plant? By photosynthesis. Where does this occur? On the leaves, the green parts, the parts which contain chlorophyll. So now, whatever sugars have been produced by those leaves, they have to be transferred from the leaf to the stem and then to the remaining part of the whole plant. So we see that the direction of water is going from root to upwards, whereas the direction of the sugars are going from upwards to downwards. Okay, so let's begin now. We see here that the veins of a leaf, they consist of xylem and also of phloem too. Alright, remember, xylem was for the conduction of water and phloem was for the conduction of the sugars. Okay, these veins of the leaf, they are then continuous with the stem of the leaf. Remember, I told you that it is always going to be continuous, especially when we're talking about the phloem. It has to be traveling, or rather the sugar which is produced from the leaf level, from the root or the tip of the leaf even. From there it has to go and transport all the way up to the remaining parts of the plants. So there has to be a system, or there has to be a network which is going to connect all the different parts of this plant, and that is the veins which are present. So the veins of the leaves are continuous with the stem of that plant. Okay, now we see here that when we're talking about xylem and phloem, let's initially talk about phloem. When we talk about phloem, whatever phloem sieve tubes which are present, they are very minute. Okay, so actually going in and studying them becomes a different task altogether. So in order to find out, or in order to see the phloem fluid, okay, or rather what are the substances which are traveling or which are being transported with the help of phloem, we take the use, or we take the help of a certain insect. Now this insect is called as an aphid. Alright, now what does this aphid do? This aphid is going to the stem, and the aphid is going to insert its proboscis. Can you see where the proboscis is? This part over here. Okay, this proboscis is inserted into the stem, and it is believed that the proboscis can go and pierce into that stem all the way up to the level of the phloem. So can you see, initially this part here is indicating the xylem, the second part inner to it is indicating the phloem. So can you see how that proboscis, means that sucking apparatus of this aphid, okay, this insect here is called as an aphid. Now that sucking apparatus of the aphid, when it is pierced inside the stem, it can reach all the way up till the level of the phloem. Okay, now what does it do while whenever it reaches the level of the phloem? Naturally, it wants food from it. So what is it going to do? It is going to pull that food out, right? So now if we want to know how exactly the phloem fluid is made up of, or what exactly is it made up of, what is it consisting of, wouldn't it help if we got whatever was in that proboscis fluid? Yes, but the proboscis is very small. So now how do we detect it? Now what happens? Let's just see over here. Okay. So basically what happens is the proboscis is the part of this aphid which is going to help us to determine what exactly juice or whatever fluid has, or whatever minerals and the sugars which has that aphid obtained from the level of the phloem. That is what the proboscis will help us to obtain. And how do we do this? First, what we'll do is we will observe this aphid right when the experiment is going on. So when the aphid is actually during the process of feeding from that plant, the aphid will have its proboscis inside the phloem. Okay, so now what do? We go and we kill the aphid. Okay, so the aphid has to actually give up its life for us to conduct this experiment. Why are we conducting this experiment? To find out what is the phloem actually made up of. Okay, so this aphid, while it is feeding on the plant, the aphid is cut, and after the aphid is cut, its proboscis part, that is the hollow part of the proboscis, is found to be penetrated all the way up till the level of the phloem. Now, as it is penetrated up till the level of the phloem, and as we have cut that aphid and only the proboscis is there inside the phloem, it behaves as an apparatus through which the phloem fluid is going to start to leak out drop by drop. So these drops can be collected, and that would be easy for us to study. So basically this aphid is killed while it is feeding, and then that hollow proboscis is still left inserted into the level of the phloem. Okay, it is found that because the contents of the phloem sieve tubes, okay, whatever contents are there in the phloem sieve tubes, because they are in a high pressure, so naturally what will happen is when the proboscis is cut, there is a lower pressure on the outside. So it will have a natural tendency to be flowing from inside the phloem through the proboscis to the exterior part. Alright. So, and that too, as I told you a little while back, that whatever phloem is coming out, whatever material is coming out of the phloem, it will be coming out in the form of small drops. Okay. Alright, now when that fluid is taken and it is studied, it is seen that that fluid is made up of sugar and it is made up of amino acids as well. Okay, so basically when the proboscis is cut, or rather when the aphid is cut from that proboscis is left inside the stem, and a pressure is created because the inside of the part of the plant is going to be having a higher pressure than that of the external part. Naturally, the substances from inside the plant is going to have a tendency to come out of the proboscis, and that is coming out in the form of drops. The drops are collected. Now, when the drops are collected and studied, it is seen that these drops contain sugars as well as amino acids. Okay, so we see here that the drops are containing sugars and amino acids, and those are all made at the level of the leaves. Don't forget that all this starch that is made in the plants, remember, by the process of photosynthesis. If you remember, there was a light reaction, a dark reaction that we did when we did the other chapter, right? So these sugars which are made, the starch that is made, has to travel and has to transport to all the other parts of the plant. So can you see here how we are indicating about the phloem here, and it is shown how the sucrose is going to go and have a downward motion, a downward movement, because it is traveling, or it is starting from the level of the leaf. So can you see here how they have shown that there is a source cell, okay, over here, that is the leaf, and also with the help of the companion cell near it, okay, the starch molecules are inserted inside the stem, and they are taken downwards with the help of this vessel which is called as the phloem. On the other hand, you can see the diagram right next to it where we are talking about the xylem. Okay, xylem is all for the transport of water. So basically when we are talking about water, water is being absorbed at the level of the roots. Okay, water is being absorbed at the level of roots, and that water has to be reaching to all the different parts of the plant, and it is the xylem which is going to take care of that. And xylem, because it is starting from the roots, we say that it has an ascending or upward movement. So can you see how here it's indicated that transport of water is taking place in an upward direction. Okay, so now we see that other experiments, in order to illustrate how sugars are being conducted, okay, when you want to talk about the conduction of sugars, remember when we talk about conduction of sugars, what do we mean? We're talking about which vessel over there? We're talking about the phloem, okay, for conduction of sugars. Now, in order to experiment or in order to show that sugars are conducted by the phloem, what we can do is, this is an experiment conducted on a bark. Okay, now on this bark what they have done is a ring has been cut. All right, can you see that here? Even in this real diagram you can see how a ring has been cut, and from the external till the center region up till the level of the phloem there is a cut being made. Okay, all the way up till the level of the phloem there is a cut being made over here. Now, what do we see here? Naturally, we know that xylem flows upwards and phloem flows downwards. So, as per natural processes, the phloem will be flowing downwards, or the constituents inside the phloem are going to be flowing downwards. But what happens here now? There is a cut over here which has been made, right? So, phloem contains the sugars which are continuously being produced at the leaves, right? So, the sugars are being produced and the sugars are coming downwards. But there is a breakage at this level here. At this level, there is a breakage. So, what happens here? All the sugars will gradually start to accumulate at this level. Okay. So, because we see that the sugars are going to start accumulating at this level, that is why we see that this level is going to be a little bulgy. Can you see here how when the ring is being cut over here, okay, after a gradual time you can see a bulge which has been formed. What is this bulge actually? This bulge is nothing but it is going to be accumulation of sugars at that region, because the sugars are not having a further place to go downwards. Okay, so if this is allowed to happen, or if this is allowed to continue on for a longer period of time, then what happens is the sugar does not flow downwards, and hence this part of the plant will not be getting any of the sugars, and also the xylem would not be flowing upwards, so the water would also not be able to go to different parts of the plant. So what happens after a given period of time, if this kind of ring is cut from the bark of the tree, then the plant would gradually be dying, and this would lead to death of that plant. Now, this is something that is, this was an experiment that we have manually done. But in the course of nature, when you observe events that are happening in forests on a day-to-day basis, okay, when you observe these kind of events, especially in the different seasons of the year, we are seeing that this kind of destruction that is there is happening naturally also due to certain animals. Okay, now there are certain mammals there, and what do? They start scratching the bark of the tree. Why do they do that? During seasons which are very cold, like the winter seasons, these animals look for food everywhere because there is a scarcity of food. Many of the other animals which they would ideally hunt and eat, they go down for hibernation. So what do these mammals do at this time? They go and they start scratching the bark of the tree, and they are going to naturally find food inside there, because they'll manage to reach, by scratching they would manage to reach all the way up to the level of the phloem. All right, so what they do, they have a general practice of doing this. Whenever they see that there is a scarcity of food around them, they go to the barks of the trees and they start scratching the barks there, and they would obtain food from the phloem. So mammals scratching the bark of the trees to get food which is stored in the phloem, that too especially during winters when the food around them is in a scarce form. So at this time, this was an example where the mammal was scratching the bark of the tree. But sometimes there are certain animals which are even so small that they are able to affect even the small seedlings which are growing. Okay, so what happens is wolves are there. They can do this to young saplings at a ground level. Not only wolves, more destructive in these forest areas would be the rabbits. Okay, so these rabbits can also be destructing all the saplings over there, and this is the reason why many of the foresters, what do they do in order to prevent this from happening? They are going to around their whole forest in order to prevent rabbits, in order to prevent these destructions happening. The fences are built all around the forest. Okay, these fences are going to be constructed all the way up to the ground level, so that no rabbit can even manage to squeeze into that forest area. All right, because the rabbits in turn, you can see that they are destroying the trees over there. They are destroying, if they're even going to go and eat those saplings, then that's going to be destroying further cropping. That's going to affect the agriculture that might be growing over there, and naturally that's going to affect the green area which is there already, the trees which are already there. That is why many foresters are opting for this method of fencing, that is the building of this net over here, and these are called as rabbit nets, so that they keep the, or rabbit proof nets, so that they keep the rabbits away. All right, so basically, even though we find rabbits to be very cute and very pleasant animals, that's not the case for these foresters. All right, now also we see that in order along with building these kind of fences and these kind of nets in order to keep these small animals away, the foresters also encourage certain predators to come. Okay, and they encourage the presence of these kind of predators in their forest, because predators such as foxes, hawks, owls, all these kind of predators are not going to commonly go and destroy the trees or the plants for their source of food. Their major source of food would be based upon their hunting. All right, so they're not going to be relying, these animals are not going to be relying upon the present plants for their food. They would be just relying upon the animals. So these animals, because of the presence of predators like foxes, hawks, owls, what do? They are going to naturally keep down the population of wolves and rabbits. How do they do that? This is what they would hunt. So if these kind of predators are there, then rabbits and wolves would naturally be staying away from this forest area, and this would help that forester prevent the destruction of any new crop that they may have laid down in that area. All right. Okay, so I hope those kind of concepts were clear, where we spoke about xylem, we spoke about phloem. Okay, we spoke about certain how transport is happening. Okay, now I'd like to come to certain terminologies for yourself. Okay, understand certain terms which will be useful when you further study about the transport, how the transport is happening. Okay, so we were gonna be learning about transport of water, transport of the minerals happening here. So let's just come across first certain terminologies. Okay, what exactly is diffusion? So let's see here, when we talk about diffusion is basically the spread of particles through random motion. Okay, like for example, if I have a perfume bottle here, and if I spray that perfume bottle, then you know that bottle of perfume in this room that I am sitting, it will, in a matter of seconds, those molecules which I have sprayed are going to manage to go in the whole room, and the fragrance would spread around the whole room. Now, have I spent any energy in spreading these these smell molecules in the whole room? I haven't. So this means that this spreading of the molecules, why has it spread? Because where I spread it near myself, its concentration was very high. So these same smell molecules have a tendency that they will have a tendency to go towards that region where there is nothing present. So they have a tendency for or moving from its area of its own higher concentration to an area of its lower concentration. Okay, and that too, this is done without energy. Okay, so spread of particles through random motion from regions of higher concentration to regions of lower concentration. Okay, then Look at this over here. You can see how the spread of molecules is happening naturally from the area of its higher concentration to the area of its lower concentration. Alright, and that what you see here, these green particles are the solute particles which are spreading. Okay, like I told you how you had the smell molecules coming from the perfume which we can spray. Okay, alright. Now, after diffusion, let's see here what other points that we have mentioned about diffusion. Diffusion of each substance is all going to happen according to its own concentration gradient. Means if it is absent at a given area, then you will see that diffusion might be happening at a faster rate. So depending on its own concentration, the diffusion happens. Okay, if there is no driving force, then there will be no diffusion at all. So in a given room, if already the odor molecules are equally spread, then you're not going to be finding diffusion happening anywhere. Diffusion will happen only when at one region there is a high concentration of a substance and at the other region there is a low concentration of that same substance. Only then that substance will be traveling from the area of its higher concentration to an area of its lower concentration. Okay. Alright. Now, after the term diffusion, I'd like you to come to the next term, and that term is called as osmosis. Okay, now these terms are not only going to help you here in transport of plants, but these terminologies are also going to help us when we study the further chapters of human physiology, further chapters of related to the human body. When we see there is a lot of functional things happening there, and these functional functions which are happening do occur by these terms of diffusion, terms of osmosis. Whenever in our body, when we're eating something, you know that absorption happens. Absorption is happening by how? Absorption happens by the process of diffusion, by the process of transfer of the molecules from its higher concentration to its lower concentration. Correct? Also, sometimes when we're talking about water, when we talk not solute particles, but when we talk about water, when water is going to be traveling from its area of its own higher concentration to an area of its lower concentration, then this is what we can call as osmosis. Okay, so when we're talking about water, the terminology that we're going to be using is going to be called as osmosis. So let's read how what the definition says. Osmosis, movement of water across a semi-permeable membrane from an area of the low solute concentration. Okay, now look at this, what it's written here. We know that we're talking always about moving from high concentration to low concentration, correct? And what are we talking about when we talk about osmosis? We are talking about water. Okay, so when we talk about water, basically what we see here is, look at this diagram. I'd like you to see this diagram. So basically here, the blue circles are indicating water and the green circles are indicating sugar. And sugar here is going to be called as the solute. Okay, and water would be the solvent. So together they are making the solution. Okay, now what is written in the definition? Let's come back to there. So movement of water, okay, remember we're talking about movement of water across a semi-permeable membrane. So here is that semi-permeable membrane we are talking about, and that too from an area of low solute concentration. Okay, we're talking about low solute concentration to an area of high solute concentration. Okay, so over here if you can see, the concentration of solutes, that is the concentration of sugar, is low. Okay, and that is moving to, what is moving? Water is moving from where sugar is low to where sugar is high. Okay, so it is movement of water, but that movement of water is depending upon what? It is depending upon the solutes present. Wherever solutes are low, water will not stay there. Water will always go where solutes are higher. Okay, in our body if you see, and if I talk about salt and water, let me talk about salt and water, very common transport in our body. Okay, wherever there is salt, naturally water is going to go behind it. Water loves salt. So naturally, without having to say, without spending any energy, water naturally and directly goes wherever salt is present. So the movement of water from its area of low solute concentration to an area of high solute concentration is going to be called as osmosis. I hope that is clear. Okay. Now, why is exactly this movement happening? Let's see that. The reason for water movement is that solutes have a tendency. Imagine if I say either I talk about sugar or if I talk about salt. The solutes have a tendency to attract water. Attracting water, loving water. So, we see that they are hydro means water. Philic means loving. So, they are water loving. Okay, if I want to say water hating or want to stay away from water, that would be called as hydrophobic. Okay, so phobic means fear, so staying away from, and when we talk about philic, it means loves. Okay, so going towards it. So we see here that the reason that water is moving is because the solutes have a tendency to attract water. Now, if there is an increase in water movement into a cell, inside a cell, if water is moving inside the cell, then we can call this type of osmosis as end osmosis. Okay, end osmosis means when water is moving inside the cell. But if at all conditions are such that water is being made to move outside the cell, then that type of osmosis is what we're going to be calling as exosmosis. Okay, so water movement outside the cell is called as exosmosis. So we not only studied what osmosis is, that is the transport of water, but we have also studied that if water enters into a cell, then that would be called as endosmosis. If water comes out of the cell, because maybe outside there is more solutes, then that would be called as exosmosis. Okay. Now, I'd also like you to come towards a terminology, and that terminology is going to be called as tonicity. Okay. Now, what is tonicity? Tonicity, let's come to that term, tonicity. Okay. When we talk about tonicity, it is all about osmolarity. Now, one more word. So, ma'am, what is osmolarity? Okay. Well, osmolarity is basically the concentration of solutes. Okay. So, let's say you have any given solution with you. How much of solutes is present in that solution is called as the osmolarity. So, osmolarity means the concentration of solutes in a given solution. Okay, and if we talk about osmolarity, how do you measure osmolarity? It is measured in the terms of milliosmoles per liter. Okay, so that is osmolarity. When I talk about osmolarity, I am only talking about one solution, but let's say now I am talking about two different solutions where one has a higher osmolarity, another one has a lower osmolarity. So that is when we come across the word which is called as tonicity. Okay, tonicity is basically the comparison of the osmolarities of two different fluids. Let's say I give an example where, let's say there are three beakers. Okay, we're talking about three beakers here. Let me draw the three beakers. Here you have beaker number 1. Okay, beaker number 2. Alright, are you able to see this? Okay, perfect. And beaker number 3. Okay, now all these beakers I am going to be filling first with water. Okay, so we'll have water and all the three beakers, and that too will have the same amount of water. Okay, and inside the beaker we will do one thing, and we will place a cell. Okay, we place a cell inside the beaker. Now what we will do is, you know that water is there and the cell is there. Okay, now let's put some salt into the water. Okay, so this is the water here. All right, let's fill all the three jars, all the three beakers are now filled with water. All the three beakers have now been filled with water, right? Now what I'm going to do is, I'm going to be adding some salt. Okay, so when I add some salt, let's see, in different beakers we'll be putting in different amounts of salts. Okay, now you know that RBC is a cell. Over here I've put the cell as a red blood cell. Since it is a cell, that cell is also going to be having certain amount of cytoplasm inside it, and the cytoplasm is also containing certain amount of salts. Okay, so now let us say that I consider however much salts are present in the cytoplasm of this RBC. If I am going to be adding the same amount of salt in this water, okay, so now I have water and I'm adding salt into it, but the salt quantity in the cell is the same as the salt quantity in the liquid or in the water, then what is this water solution called as? Yes, so I have two things here. Remember, tonicity means two solutions. I have my two solutions. Which are my two solutions? My first solution is the water solution, and my second solution is the cell or the RBC. Okay, now RBC is a cell, it has cytoplasm, and cytoplasm has salts, and we've also put salts in the water, but we've put equal amount. So can I say that in this beaker number 1, the solution that is there, okay, I'm talking about the water solution, that solution is an isotonic solution. Okay, why is that an isotonic solution? Because the concentration of salts in the solution is the same as the concentration of salts in the cell. Did you understand this? Okay, now next beaker number 2. Okay, let us say now however much concentration of salt is present in the RBC. Okay, there is a given amount of concentration of salt in this RBC, but I am going to put very little salt in my water solution, very little salt. Okay, so naturally what is happening here? Salt in the water is going to be less than the salt present in that RBC, and because salt in the water is less, salt in the water solution is less. So this solution here can be called as a hypotonic solution. Okay, the solution is called as a hypotonic solution because salts in the water solution are less as compared to salts which are present inside that RBC. Okay, now let's come to the third beaker. In this beaker also, we know that there is going to be a given amount of salt which is present inside this RBC, okay, given amount of salt present here, and let's say I am adding salt to the third beaker in that water solution, and let us say that I add too much of salt, excess of salt I am going to be adding. Okay, I am adding how much? I am adding excess of salt. Can you see, as compared to the first two beakers, I am adding way too much salt over here. Okay, so now the presence of this salt here is much more than the salt which is present inside the RBC. So now which solution will this be called as? Because considering with the terminology of the tonicity, okay, I am talking about tonicity here, this is going to be called as a hypertonic solution. Okay, this is going to be called as a hypertonic solution. Now, since we have now identified the three different types of solutions, let us also go ahead a step and understand what is the changes that's going to happen of the RBC over here. We learnt about osmosis, didn't we? We learned that osmosis meant that water is going to go from its area wherever there is going to be low solutes, it will go to the area where there are high solutes. Okay, so now let's see the comparison here. When we talk about beaker number 1, where is the solutes higher? There is no difference because it is a isotonic solution. When we say it is an isotonic solution, so salts in the cell are same as salts in the water solution. So there is no difference there. So there will be no movement of water. But when we talk about this hypotonic solution, okay, in the first one there is no movement of water. Okay, so what happens to the RBC over here? The cell or that RBC remains the same. Okay, now when we talk about this other solution, the next solution is indicating a hypotonic. When I say that the solution is hypotonic, I mean that there is going to be more solutes inside the cell. So, more solutes inside the RBC. So now, if there is more solutes inside the RBC, where is the water going to go? Water goes wherever there is more solutes. So naturally, water is going to go inside the RBC, and that will cause that RBC to enlarge. Okay, so that is the reason why we are going to be seeing that there will be an enlargement in the size of this RBC, and this enlargement that we are seeing in the size of the RBC, it is said to be that the RBC is going to become turgid. Okay, it is going to swell up. Alright. Okay, so there is more solutes inside the RBC and the RBC swells up. Okay, or you can say that it becomes turgid. Alright. When we talk about the next solution, that is a hypertonic solution. What happens to the hypertonic solution? Remember, hypertonic means that there is going to be hypertonic solution, means there is going to be more solutes in the solution. So what's going to happen? Water is going to go irrespective of anything. Water will go to the area where there's going to be more solutes. So naturally water will be going from inside the RBC into the water solution. Okay, so what happens to the RBC? Then what happens we see here is the RBC is going to shrivel. Okay, it's going to become very small. So the size of the RBC reduces. Okay, and all the water which was there inside the RBC cytoplasm has ended coming back out, and because outside there is a lot of salt. Okay, so irrespective of anything, the water comes out and the RBC just shrivels down. Okay, so this is also, you can call this as, it is the RBC will shrink, or you may say that it becomes flaccid. Okay, so the third experiment shows that RBC becomes flaccid. Okay, or you can also call this as RBC becoming plasmolized. Okay, so now we studied about the different types of ways that water can be transported. Okay, we learnt about diffusion. We also learnt about osmosis. So these are two terminologies which are going to be helpful when we're talking about transport happening anywhere. Okay, remember, these are all done without the help of energy, okay, being utilized at all, because of the natural tendency of water to be moving all these substances. Okay, so that was that. Now just same thing that we just studied a little while back. Okay, let's see that here. What exactly is a hypertonic solution? It is a solution which is having hypertonic. Okay, we're talking about hypertonic. Remember what was tonicity? Tonicity meant the concentration of solutes. Okay, so hypertonic solution A solution having more of a solute concentration, more of solute. That's why hypertonic. If the cell is placed in such a solution, then water is going to be moving out of the cell and goes into the solution, like we spoke about the RBC. When the solution is having too much of solutes, water comes out of the cell and the cell becomes shrunken down. And that movement of water out of the cell, we learned that it was called as, when water moves out of the cell, we call that as exosmosis, right? So that movement of water outside the cell, because there are too much of solutes outside the cell, it's called as exosmosis. After hypertonic solution, let's move on to hypotonic solution. In hypotonic solution, these are those solutions which are having a less concentration of solutes, okay? So there is less solute concentration, and because the concentration of solute in the surrounding solution is less, so there is more concentration inside the cell. That is why water is going to have a natural tendency to flow from the outside to inside the cell. And because water flows inside the cell is going to swell up. And movement of water inside the cell is going to be called as endosmosis. What is it called as? It's called as endosmosis. Okay. Then our third type of solution will be that kind of solution where the amount of solutes in the solution of water and the amount of solute inside the cell are exactly the same. So when they are the same, remember I told you that osmosis is always depending on its own concentration. So if the concentration is the same in both the areas, there will be no movement of water at all. That's why we are going to be calling this as a isotonic solution, where both the solutions are in equilibrium. And over here, then there will be no movement of water. So considering our cell experiment, the water inside the cell is going to be the same, and there will be actually no movement or no changes happening at the level of the cell. Okay, so I hope this cleared out your doubts about diffusion, about osmosis. Remember, diffusion was all about solute particles and osmosis is all about the water molecules. Okay, now, well, we have almost come to an end to this chapter of transports. Okay, in our next following lectures, we're just going to be doing some very interesting topics about some blood related disorders, and before that we're going to be learning about how exactly do we determine the different blood groups which are present in our body. Okay, we're going to have a brief insight about that, and we're also going to be stepping into how exactly is blood transfused. Okay, you know that sometimes certain people cannot donate blood to certain other people. Why? Because the blood groups don't match. So when I'm saying that blood groups don't match, what exactly are we meaning by blood groups not matching? Okay, so this is what we're going to be learning in our very next lecture next week. Alright, now I'd like you all to let me know whether you found today's lecture helpful. What you can do is, you can, if at all there are any doubts in today's lecture, you can go to our platform where you can download the Yupp Master app, and in that app we have a 24/7 live doubt session. So children, whatever doubts you would be posting on that, I could personally handle your biology doubts if at all. And also I'd like you to like and comment on any of these videos if you found them helpful. And please do, if you found this video helpful for you all, please do subscribe to our channel. See you in the next lecture. Till then, you guys stay home and stay safe. Bye.